Greetings! Welcome to my channel, Human Evidently, where I provide evidence-based videos on human origins, paleoanthropology, archaeology, and evolutionary science for adults. They say the best way to learn is to teach, and as an anthropology student, I have found this to be true. My presentation videos are developed from the study material I build from my undergraduate courses and scholarly sources, and can be used to supplement your own learning in high school or college level classes. This video is part of my ADHD-friendly study guide series. Today we will be going over a study guide for Week 1 of ASM 443, Fossil Hominins. Fossil Hominins is an online lab and lecture course at Arizona State University. I attended this course in spring of 2026. Today we are covering the readings from Week 1. This video is made based on the presupposition that you, the viewer, have already read the course materials and are wanting to review the material before taking your test. If you try to use it without having read the material first, you will not get much mileage out of this presentation. Before we dive straight into this week's readings, let's do a quick recap of what you need to know this week. Macroevolution is evolution at or above the species level. Speciation, extinction, diversification, and large-scale trait change. The origin of the human lineage is therefore a macroevolutionary question, though it was built from microevolutionary processes within ancestral populations. Evolution is change in heritable characteristics across generations. Microevolution is that change within populations via mutation, selection, drift, and gene flow, so it is part of evolution, not a different phenomenon. Macroevolutionary patterns include stasis, anagenesis, cladograms, adaptive radiation, convergence, and extinction. A species is often defined as an independently evolving lineage. Major concepts include biological, morphological, phylogenetic, and ecological species concepts. Speciation occurs when gene flow is reduced and reproductive isolation evolves. Classification and taxonomy are grouping systems. Phylogeny is the history of relationships. We reconstruct phylogenies to infer ancestry, timing, trait origins, and test hypotheses about function or behavior by comparing relatives. Classification should reflect evolutionary relationships, not superficial similarity. A cladogram shows branching order only. A phylogenetic tree may also show amount of change or time. Gradualism predicts more continuous change. Punctuated equilibrium predicts long stasis with geologically rapid change near speciation. If all of that made sense, congratulations! You don't need to watch the rest of this video. You can if you want to, but if you were tracking all of that, you likely have a solid grasp on this week's concepts. That said, if you want a more in-depth coverage of the topic at hand, keep watching. The first source we're covering is the Nature Citable article, Evolution is Change in the Inherited Traits of a Population Through Successive Generations, by Forbes and Krimmel, 2010. The label on this box states exactly what you'll find inside. The definition of evolution is change in the inherited traits of a population across generations. Contrary to an unfortunately common belief, evolution does not happen in individuals. Instead, it happens in populations of individuals. No one individual organism undergoes evolution during its lifetime. Instead, inherited traits are ultimately tied to genetic information passed from parents to offspring. When genetic sequences in a population change, and those changes are inherited, that is evolution. Traits can be obvious and have a phenotypical presentation, such as eye color or wing patterns, or they can be hidden within DNA sequences. Biology, including ecology, is best understood through an evolutionary framework, because evolution helps explain how organisms interact with their environments. Ecology makes more sense when we know that populations are constantly shaped by inherited change over time. What is evolution not? Evolution is not directional or goal-directed. It does not try to become perfect or more advanced. Evolution is not an entity that makes decisions at all. It is a process that occurs naturally due to pressures in the environment. Nor is evolution a synonym for progress. Evolution can lead to increasing complexity as much as it can lead to decreasing complexity. Terms like devolution or reverse evolution are misnomers for the same process of change over time in response to environmental pressures or lack thereof. Natural selection is one mechanism of evolution, but it is not the only one. Trait changes in populations are not always caused by selection. Some genetic changes may be neutral, meaning they do not affect fitness much or at all. There are two scales of evolution, micro and macroevolution. Microevolution is evolutionary change within a species. This includes changes in trait frequencies, or allele frequencies, in a population. It can happen over relatively short spans of time. Macroevolution is evolutionary change above the species level. It often includes speciation, where populations become unable or unwilling to interbreed. Because macroevolution happens over long periods of time, scientists often study it through fossils, phylogenies, and long-term patterns, whereas microevolution is more likely to be observed in nature or a lab setting in real time. So, in summary, the theory of evolution provides the framework for understanding biology and ecology because it elucidates the change in inherited traits of populations over generations, driven by multiple mechanisms. It does not imply progress toward perfection, and its mechanisms are not solely relegated to natural selection. Naturally, the next question is, well, what are the other mechanisms? If you already studied, you know that this is addressed by the next article, titled Natural Selection, Genetic Drift, and Gene Flow Do Not Act in Isolation in Natural Populations, by Andrews, 2010. Natural selection, genetic drift, and gene flow are all mechanisms of evolution because they can change allele frequencies over time, and, i.e., in real populations, these mechanisms usually work together, not one at a time. When these forces act, the population no longer fits all the assumptions of Hardy-Weinberg equilibrium, so evolution is occurring. For natural selection to occur, three things are needed. Variation exists in a trait, the variation is heritable, and the variation affects fitness. Then, some genotypes survive and reproduce better than others, which causes their alleles to become more common in the population. Within the umbrella of natural selection is directional selection. Directional selection favors one allele, so that allele increases over time. If selection stays consistent, a favored allele may eventually become fixed in the population. But how likely are these alleles to spread? Well, there are dominant and recessive advantageous alleles. A rare dominant can spread quickly at first because it shows its effect even in heterozygotes. A rare recessive spreads more slowly because it is less likely to appear in homozygotes. Interestingly, this also makes harmful recessive alleles more resistant to eradication. Sometimes, alleles only show fitness when complementary alleles are present within a population. This is called balancing selection, which keeps multiple alleles in a population instead of pushing one to fixation. One way this happens is heterozygote advantage, where heterozygotes have a higher fitness than either homozygote. Similarly, negative frequency-dependent selection can lead a genotype to be less fit if it becomes too common. Other evolutionary mechanisms are genetic drift and gene flow. Genetic drift is random change in allele frequencies caused by chance, and it comes from basically a sampling error when only a limited number of gametes are passed to the next generation. Drift is strongest in small populations, and it can cause an allele to become fixed or lost purely by chance. It tends to reduce genetic variation within populations and increase differences between populations. Gene flow is the movement of genes into or out of a population. It can happen through the migration of organisms or a movement of gametes, as in pollen. Gene flow usually makes populations more genetically similar to each other. When gene flow is limited, populations can diverge more through selection and drift, which can contribute to speciation over time. One key component to gene transmission is population size. Effective population size is the number of breeding individuals contributing genes to the next generation. Effective population size may be smaller than the total head count because of unequal sex ratios, mating structure, or changes in population size over time. Real populations are finite, so genetic drift is always potentially present. In small populations, drift can overpower weak selection, so even a beneficial allele may fail to spread. Gene flow can also oppose local adaptation by continually introducing alleles that are useful elsewhere, but harmful in the local environment. The interplay of all these forces leads to the eventual change of allele frequencies over time. The impact of the interactions of these forces matter a lot in conservation genetics. Species living in small, fragmented populations are more likely to be pushed in different directions than larger populations. Small populations are vulnerable to losing heterozygosity and fixing harmful alleles through drift. Population bottlenecks can have long-lasting effects because short periods of low population size can greatly reduce effective population size. Interestingly, about one migrant per generation can be enough to counteract the diversifying effects of drift among subpopulations. Ultimately, natural selection, genetic drift, and gene flow all change allele frequencies. They interact in real populations to shape adaptation, diversity, and population divergence. So evolution means populations change over time, and evolution happens because of natural selection, genetic drift, and gene flow. But what about mutations? The next article is titled Mutations Are the Raw Materials of Evolution by Carlin, 2011. According to this article, which covers a topic that's pretty solid to the foundations of the science, a mutation is a change in the DNA sequence of an organism. Mutations happen, and natural selection selects for or against that mutation. When mutations are selected for, they can lead to new genetic variants. Mutations are typically caused by environmental factors or coding mistakes during DNA replication. Some mutations appear spontaneously, even when nothing obvious in the environment caused them. The main types of mutations, at least for the sake of this course, are point mutations and chromosomal aberrations. A point mutation changes DNA at a small scale, often involving one base pair. Common point mutation types include substitution, deletion, duplication, and insertion. Chromosomal aberrations are larger scale mutations that affect bigger pieces of DNA. They can happen during meiosis, unequal crossing over, DNA recombination errors, or because of transposable elements. Entire genes or even whole chromosomes can be duplicated, deleted, or rearranged. DNA copying is usually very accurate, but mistakes still happen. Changes occur at roughly 1 in every 10 to the 10th base pairs copied. These mutations can be harmful, neutral, or beneficial. Many are harmful or have little noticeable effect. Very rarely, a mutation will increase fitness and becomes an adaptation. When understanding mutations, it's important to know where the mutation occurred. Somatic mutations happen in the body cells and are not passed on to offspring. Germline mutations happen in gametes and can be passed on to future generations. Every inherited genetic feature ultimately can be traced back to some past mutation. A new allele produced by a mutation can spread, changing fitness and influencing evolutionary change over time. This article uses snake venom as an example of how many small mutations can gradually produce major evolutionary differences. Slight mutations affecting salivary enzymes accumulated over time, and in snakes, these changes contributed to venom evolution. Different snake lineages ended up with different kinds of venom because mutations were random and selection favored different variants in different populations. Evolution is not always slow, though. Sometimes a mutation can produce very large changes quickly. The gray tree frog example shows chromosome doubling, which produced a new form with 48 chromosomes instead of 24, where the frog looked nearly identical but had a new mating call. This is a case of very rapid or near-instant speciation through genome duplication. In summary, mutations are changes in DNA that create new genetic variation, and that variation is the raw material of evolution. Now we're getting to the good stuff. Let's talk macroevolution in primates with the article Macroevolution: Examples from the Primate World by Klee and Gonder, 2012. Macroevolution looks at large-scale evolutionary patterns above the species level and across very long time spans. It includes stasis, adaptive radiation, extinction, convergent evolution, and speciation patterns. The article contrasts macroevolution with microevolution, which focuses on allele frequency changes within species through mechanisms such as mutation, migration, drift, and selection. Fossils and phylogenetic trees are especially important for macroevolution because they help show broad changes in lineages over time. So let's talk about what that looks like. Macroevolution helps explain both the diversity of life and the tempo of evolutionary change. Phyletic gradualism is when species change gradually over long periods of time. Punctuated equilibrium is when relatively rapid branching or speciation happens, often followed by long periods of little change, known as stasis. In punctuated equilibrium, major environmental changes can open new niches or trigger extinctions, which can reshape the tree of life quickly. The major macroevolutionary patterns in primates are stasis, adaptive radiation, extinction of lineages, convergent evolution, and anagenesis. Large primate phylogenetic studies based on genetic data show that primate evolution includes both long-lasting relic lineages and bursts of diversification. Tarsiers are used as an example of a likely ancient relict lineage, with a long branch suggesting relative stasis compared with other primates. Lemurs are used as an example of likely adaptive radiation. Many short, early branches suggest rapid diversification, likely associated with the colonization of Madagascar around 62 to 65 million years ago. New World monkeys split from Old World monkeys roughly 30 to 50 million years ago, but their later diversification is complex. They show a combination of early radiation, a period of stasis, and later extinction of many early lineages, followed by another burst of diversification into modern crown lineages. This matters because macroevolution is often not one smooth process. Lineages can radiate, stabilize, collapse, and radiate again. During the Miocene, ape and Old World monkey ancestors experienced both radiations and extinctions linked to climate change. In the early and middle Miocene, warming and the spread of tropical habitats helped apes expand into Eurasia, producing a major diversification of ape species. Later, a shift toward drier, more open habitats contributed to a major decline in Eurasian hominoids. By about 5 million years ago, most of those ape species were gone, leaving only a few lineages tied to living apes such as orangutans and gibbons. A possible case of anagenesis is the transition from Homo heidelbergensis to Homo neanderthalensis in Europe. Genetic comparisons suggest that Neanderthals and modern humans likely shared a last common ancestor, probably H. heidelbergensis, around 0.35 to 0.40 million years ago. Some researchers interpret the fossil record as showing a gradual buildup of Neanderthal traits over time, which is why some paleoanthropologists argue for an anagenetic transition. The article also uses primate color vision to show convergent evolution. Most mammals are dichromats, while many primates, including humans, are trichromats, with 3 visual pigments. In most New World monkeys, males are dichromats and females may be dichromats or trichromats because of X-linked opsin variation. Howler monkeys are unusual. Both sexes can be trichromats because of a recent duplication of the ML opsin gene. This is treated as a reinvention of trichromacy, and therefore as a useful primate example of convergent evolution. It's important to note that micro and macroevolution are not completely separate, and that evolution is not one, steady, simple progression. Macroevolution in primates is visible in broad patterns such as adaptive radiation, stasis, extinction, anagenesis, and convergent evolution, all of which help explain how primate diversity developed through deep time. Next up is Tempo and Mode in Evolution by Fitch and Ayala, 1994. Tempo is the rate of evolution, and mode is the pattern or manner of evolution. It's important to know how and why rates speed up or slow down over time. Human evolution is a great example of mode. Evolution can be uneven and patchy across different anatomical systems. Human evolution is mosaic, meaning different traits changed at different times rather than all at once. Bipedalism appeared early. Large brain size evolved later. Some locomotor, dental, and facial traits changed on different schedules. Paleontology and genetics combine to help illuminate the modern evolutionary synthesis. The historiography of the theory of evolution is useful to know here. Darwin argued that evolution usually happens through the accumulation of small differences over time. Some others, like Huxley and Galton, argued instead for more sudden, discontinuous changes, or evolutionary jumps. The conflict between biometricians and Mendelians was resolved when population genetics showed that Mendelian inheritance and gradual evolution could work together. Dobzhansky's Genetics and the Origin of Species helped bring the theory together with real biological evidence. Simpson treated evolutionary rates as outcomes of genetic change in populations. Simpson emphasized factors that influence evolutionary rate: variation, mutation rate and effect, generation length, population size, and natural selection. Populations do not always need to wait for a new mutation because existing variation can already be present as a stored resource. Simpson also argued that selection is creative, not just destructive, because selection changes which combinations of traits and genes actually appear in populations. Simpson's work covers mode and tempo in depth, so it's foundational to more modern contributions to evolutionary theory. Simpson talks about how evolution does not always proceed at one constant speed, and the different speeds are bradytelic, horotelic, and tachytelic, respectively. He also recognized long periods of stasis, seen in living fossils, where groups change very little over immense spans of time. At the same time, he accepted that some major transitions can happen relatively quickly under special conditions. Unusually fast evolution can happen when new niches or adaptive zones are invaded. Reduced population numbers can also be associated with these periods of rapid change. This helps explain why the fossil record sometimes shows discontinuities, not only because fossils are missing, but sometimes because real evolutionary change was unusually rapid. Simpson discussed both microevolution and macroevolution, and many modern paleontological themes were already present in his work. One major issue is whether large-scale patterns can be fully explained by ordinary population-level processes, or whether macroevolution sometimes requires extra theory. Stephen Jay Gould argued that some phenomena, such as punctuated equilibrium and mass extinction, may require specifically macroevolutionary explanations. Let's cover some examples. The earliest life history is mostly reconstructed through molecular biology because fossils do not preserve the pre-cellular stages. Prokaryotic microbes more than 3 billion years old appear to have evolved very slowly. Eukaryotic microfossils appeared later, and their rates of diversification increased over time, especially near the Cambrian. The Cambrian explosion is presented as a major example of rapid macroevolutionary novelty. The older idea that this was only an artifact of a poor fossil record became less convincing after more Precambrian fossils were found. Many major body plans really did appear rapidly. So what about mass extinction? This is considered a major evolutionary force. There have been 5 on Earth that we know of so far. Ask me about my favorite in the comments under this video, and maybe I'll make a presentation about it. Mass extinctions can restructure the biosphere by wiping out dominant groups and allowing other lineages to diversify. Extinction in general is often not generally caused by natural selection. Simpson thought measuring genetic change directly through time was not practical, but molecular biology later made this possible. DNA and protein comparisons transformed the study of evolutionary rates and relationships. Molecular clocks are complex and must be interpreted carefully because rates can vary by lineage, gene region, and type of site. Experimental evolution in E. coli shows that adaptation can happen rapidly at first and then slow down. Even populations that begin identically and evolve in identical environments can follow different trajectories. Chance events can play an important role in evolutionary outcomes. So if there's a gap in the fossil record, does that mean the fossil record is incomplete? Not always. Remember, sometimes evolution can happen rapidly. Finally, let's cover homology and hominid phylogeny. This 6th source is Homology and Hominid Phylogeny, Problems and Potential Solutions by Lieberman in 1999. Note the date on some of these papers. It's important to look back on older literature while maintaining an awareness of recent and relevant sources. Lieberman's work here asks how we can reconstruct hominid evolutionary relationships from bones, when bone is often a messy and unreliable source of phylogenetic information. DNA is generally better than morphology for phylogeny, but for fossil hominids, we often do not have DNA, so we still depend heavily on skeletal evidence. Fossil hominid DNA is rare and usually only preserved in colder environments, such as permafrost, for up to about 700,000 years. While DNA has been successfully extracted from Neanderthals and some archaic humans, many older hominins, like Australopithecus, dating back millions of years, have no DNA available. Because most hominid fossils are too old or degraded, paleontology still heavily relies on skeletal evidence, like teeth, skull shapes, and bones, to understand evolution, diet, and movement, although recent advances can now find DNA in sediments. Ancient DNA is referred to as aDNA. Morphological data from bones is the primary source of information for the vast majority of fossil specimens spanning millions of years. Skeletal similarities can result from common ancestry, convergence, shared development, or environmental influences, so not every similarity is useful for building a tree. So why are bones difficult phylogenetic evidence? Bones are plastic tissues. They change during life in response to growth, function, and environment. Skeletal traits often have limited heritability and are part of integrated functional systems, which makes it hard to divide them into neat, independent phylogenetic characters. Morphology may work better for higher taxonomic levels, but it becomes less reliable for closely related species, such as fossil hominids. Can morphological data be misleading? Absolutely. Remember earlier when we talked about convergent evolution versus derived traits? The human-chimp-gorilla trichotomy shows how morphology can mislead us. Many morphological similarities seem to group chimpanzees and gorillas together, but molecular evidence shows that humans and chimpanzees are closer relatives. If morphology can mislead us about living apes, whose relationships are known from molecules, it can also mislead us about fossil hominids. So then, what makes a good phylogenetic character? A useful character should be a natural unit. It should be discrete and/or quantifiable. It should vary more between taxa than within taxa. Its derived versus ancestral state should be identifiable. It should be as independent as possible from other characters. It should be heritable. And it should compare the same kind of unit across taxa, which leads us directly to the problem of homology. In the broadest sense, homology is a hypothesis about similarity, but in evolutionary biology it is often defined as similarity due to common ancestry. That is useful in theory, but difficult in practice for fossil hominids, because it assumes you already know the phylogeny well enough to tell which similarities come from shared ancestry. Lieberman distinguishes among phylogenetic homology, morphological homology, and developmental homology. The human chin is used as a case study to show how tricky homology can be. Some Neanderthals and some early modern human fossils show partial or ambiguous chin-like projections, making it hard to decide whether these are true homologous chins, transitional forms, signs of interbreeding, or simply coincidental misleading similarities. Put simply, a trait can look similar without meaning the same thing phylogenetically. It follows, then, that a major problem in hominid phylogeny is character conflict. Different characters support different evolutionary trees. In one cladistic analysis, only about 68% of the characters supported the most parsimonious tree, meaning around 30% conflicted with it. This happens because some similarities are homoplasies rather than true phylogenetic homologies. Homoplasy means a similarity evolved independently, such as through convergence or parallelism. A standard response to character conflict is parsimony. Choose the tree that requires the fewest evolutionary changes. Parsimony is not enough, because it assumes that homology is more common than homoplasy, and that assumption can fail. If morphology grouped chimps and gorillas together, parsimony alone would have pointed to the wrong tree. So what approaches are there to characterizing species? While we didn't cover all of these in this particular paper, the approaches are as follows. The biological species concept, BSC, which defines species as groups of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. The phylogenetic or cladistic species concept, which defines a species as the smallest diagnosable cluster of organisms that shares a common ancestor and forms a single branch on the tree of life, a monophyly. The morphological or typological species concept, which defines species based on groups of organisms that share a similar physical phenotype that is distinct from other sets. The evolutionary species concept, which defines a species as a single lineage of ancestor-descendant populations that maintains its independent evolutionary identity, tendencies, and historical fate. The ecological species concept, which defines a species as a lineage that occupies a distinct ecological niche, adapting to a specific environment in a way that separates it from other lineages. The unified species concept, which suggests that many other definitions are actually just methods of identifying a more fundamental concept, separately evolving metapopulation lineages, and the evolutionary significant unit concept, which is used in conservation to describe a population of organisms considered distinct for the purposes of protection. Defining a species can involve using several methods to assess a fossil: morphology, genetic sequencing, radiometric dating, ecology, and parsimony. Then, comparing results across specimens to determine a fossil's place on the tree of life. If you find a fossil situated in the same geographic and temporal region as a morphologically similar fossil within the same geographic region, but a different temporal region, you might ask, how are these two related, if at all? And then the question becomes how to determine their degree of relatedness. I think the unified species concept is the most useful because it takes into account all of these components. The struggle, of course, comes with determining the weight of certain morphological features, genetic relatedness, and whether certain species interbred. Access to genetic material is necessary to determine these things with certainty, and aDNA is hard to find on hominin fossils older than 700,000 years, roughly. This contrasts significantly with modern populations, which can more readily be observed changing over time. Genetic testing is great, but one can witness copulations and births fairly reliably, and humans have kept written records of such things for almost as long as we've had the ability to write. In paleoanthropology, morphology is usually the starting point, but morphological and typological approaches, while practical in the absence of behavior, reproduction, and genetics, might miss things like variation in sex, age, or pathology. Additionally, as it becomes more evident that transitional species make up the majority of species because of those slow changes over time that produce evolution from one species to another, over-reliance on morphology may lead to invalid divisions between groups that don't need to be considered different species due to their general suite of characteristics, including their chronology, geography, and phylogenetic placement. A DNA is not always available, but I think the field is finding ways to navigate that issue. More DNA is available now than has been in the past due to refined tech capabilities. Case in point, the 2022 Nobel in Medicine was awarded to the Swedish geneticist Svante Pääbo for his research in the field of genomes of extinct hominins and human evolution. Things like hybridization, asexual reproduction, ring species, and taxonomic inflation add to the challenge of defining a species, which is what lends to such different yet similar schools of thought. In this course, during week 1, we covered historical versus developmental approaches. Historical, or phylogenetic approach, defines similarity strictly as similarity from common descent. Unfortunately, this can become circular because you need a correct phylogeny to identify homology, but you also need homology to build the phylogeny. You're basically building the bridge while you're standing on it. The developmental approach looks at the developmental processes that generate the trait. It is logically stronger for fossils because it asks what mechanisms actually caused the similarity. There are two developmental approaches, ontologic homology and mechanistic homology. Ontologic homology compares the sequence of development across taxa. Similar ontogenies do not always mean true phylogenetic homology, and different ontogenies do not always mean non-homology. Mechanistic homology focuses on actual proximate mechanisms of growth, such as cellular activity, timing, and differentiation. This is generally a better way to evaluate whether a skeletal similarity is meaningful for phylogeny. Given all that information, similarities are more likely to be developmentally meaningful if they arise from the same growth processes, not just the same final shape. Bones grow through a few basic processes involving osteoblasts and osteoclasts, acting in depository or resorptive growth fields. One of Lieberman's main proposed solutions is to study the mechanisms of skeletal growth, not just the adult morphology. For example, orthognathism can be misleading. Orthognathism means the lower face does not project far beyond the upper face. It appears in both Homo and robust Australopithecines, which could look like evidence that they are closely related. But these similar-looking faces arise by different developmental mechanisms. In Homo, facial orthognathism is linked to resorptive growth fields on the anterior maxilla, while in robust Australopithecines, it reflects a different pattern, including relative upper face elongation. Therefore, the similarity is not developmentally homologous and should not be treated as evidence of close shared ancestry. Traits like bone thickness and robusticity are especially risky for phylogeny because bones respond strongly to mechanical loading and environment. Examples from exercise and biomechanics show that bones can become thicker or thinner based on behavior and loading. So traits like thick cranial vaults or large crests may reflect epigenetic or mechanical responses, not reliable phylogenetic signals. So to wrap all that up, paleoanthropologists need fewer but better characters, not just larger lists of morphological traits. Good characters should be tested for heritability, independence, derivation, and especially developmental homology. Until fossil DNA becomes more available, the best path forward is to integrate phylogenetic analysis with developmental biology and functional morphology. Similarity may reflect convergence, parallelism, or shared developmental or mechanical effects, rather than common ancestry. Parsimony can fail because it assumes homology is more common than homoplasy. Similar developmental sequences can still mislead. Mechanistic developmental evidence is stronger. Bone thickness and robusticity are heavily influenced by mechanical loading and environment. And, lastly, reconstructing hominid phylogeny from bones is difficult because many skeletal similarities are misleading, so the best approach is to use fewer, better tested characters and to evaluate them through developmental mechanisms rather than appearance alone. In conclusion, our readings talked about the mechanisms and patterns that work across different scales of time to produce evolution. Taken together, these ideas help explain why the fossil record looks the way it does, and why reconstructing evolutionary history can be so challenging. In paleoanthropology especially, scientists must weigh evidence carefully, because morphology can reflect shared ancestry, but it can also reflect convergence, developmental constraints, or environmental pressures. The main takeaway is that evolution is both dynamic and complex. It can be gradual or rapid, predictable in some ways, and shaped by chance in others. By studying tempo, mode, mutation, population processes, and homology together, we get a fuller picture of how life changes through time and how our own lineage fits into that larger story. Thank you for spending this time with me on Human, Evidently. I hope you're leaving with a clearer understanding of the subject and a deeper curiosity about the story of life, evolution, and our place within it. Until next time, keep asking those good questions. If you want to support my channel, please consider subscribing here and on Patreon, where you can access my content for free or contribute a small donation to get your name at the end of my videos. I am working on a series of educational material for middle grade readers and 2 science fiction novels. Such endeavors are only made possible by contributions from viewers like you. Be sure to check out the sources in the description for more learning.